We discussed previously the need to get proteins into solution and how to go about that. Once proteins are in solution, they need to be fractionated. The main reason to fractionate is what is referred to as the dynamic range of protein concentration. Or put simply, the difference between the highest abundance protein in a sample and the sample with the lowest number of molecules, as shown in this diagram showing the concentration of proteins in plasma. In spite of the latest developments in mass spectrometry claiming to provide full proteome coverage in less than a day, the need to reduce sample complexity is still very real. There are many reasons for this, such as allowing a more reliable identification of low abundance proteins, especially proteins that are post-translationally modified, as it may only be a small proportion of the population of molecules is modified, and thus their number may not be great enough to detect. Proteins can be fractionated by their chemical properties, the three being their charge, their size, and whether or not they like or hate water, or their hydrophobicity. Affinity methods can also be used to selectively isolate proteins with certain modifications, but we'll discuss this at another time. Fractionation in this case doesn't mean isolating a single protein isoform to hit homogeneity, or only having molecules of a single isoform. We'll discuss that as well another time. Fractionation is about reducing complexity. But why do we need to reduce complexity? Simply put, cells just don't contain one molecule of a particular protein, but lots of molecules to perform reactions and functions most efficiently. Molecules of the same protein can be made or broken down and recycled depending on the cell's needs. This diagram shows what is called the dynamic range of protein concentration, or more simply, how much of a particular protein is present in a cell on average. As you can see, the range of concentration is staggeringly huge. What this means from a proteome analysis point of view is that in an unfractionated sample of something like serum, 95% of the mass of the protein is only 22 different protein species, while the remaining 1% of the protein mass is greater than 10,000 protein species. Without some kind of fractionation, such as these electrophoretic gels of unfractionated serum showing only about 100 protein isoforms, our analysis of the proteome is grossly underrepresenting the reality and thus not allowing us to make meaningful interpretations. So now that we know what proteome complexity is and the problem that it causes, how do we overcome it? First, we're going to initially restrict the discussion to a denatured proteome rather than fractionating protein complexes, which is a little bit more difficult. A denatured sample has been treated with chaotropes and or surfactants to try and solubilize as many proteins as possible. Protein size is the simplest property to use for fractionation, and electrophoresis, or moving molecules in an electric field, or chromatography, pumping the molecules through a liquid stream through a column of beads, are the two real choices for denatured proteins, and both use solid supports of either a gel or a column. We'll discuss these techniques in greater details at a later time. At this point, it is enough to know that these techniques sieve proteins by their size, allowing them to be fractionated into a few or a great many fractions. As in this example, where a sample has been fractionated by gel filtration chromatography, and those fractions have been further assessed by performing gel electrophoresis. And you can see the size of the proteins changing with the different fraction number. Charge is the next property that can be used for protein fractionation, but it's a little bit more difficult than separating by size. Firstly, the sample needs to be free of charged contaminants or anything that is not protein. The main consideration here is the surfactant used. While STS is extremely useful for solubilizing a great diversity of protein molecules and is the key ingredient in electrophoresis for separating proteins by size, its strong negative charge means that all the protein molecules have the same charge and can't be separated by their charge. For a charge-based separation, we need to employ zwitterionic surfactants that have both a positive and a negative group balancing so that the overall charge is zero and the surfactant molecule can't move in the electric field. Proteins can also act as zwitterions. as they have positively charged and negatively charged amino acid, but when balanced, the overall charge is 0. By altering the pH, the charge on the protein can be changed, and this can be used to separate different proteins. This example shows how the surface charge of 3 different proteins can be altered by changing the pH of the solution that they are in and allowing them to be selectively separated from one another. Which brings us to how proteins are separated by charge using electrophoresis. In a tube, we load a solution of proteins that we want to fractionate, dissolved in urea, thiourea, and surfactant. We add a solution of amphiolites, small molecules containing both acidic and basic groups that at certain pH are neutral. When placed in an electric field, the amphiolites can move and establish a pH gradient along the tube of liquid, as shown by the movement of the balls in this animation. Protein molecules are also amphiolites, and having amino acids with both acidic and basic side chains, the balance of these acidic or basic amino acids determines at what pH the protein's overall charge is 0 and thus can't move in the electric field. In an example like this, liquid isoelectric focusing, all of the molecules of a particular protein isoform will move to the same pH, but you can only have so many molecules in the same piece of space. So the higher the concentration of the protein, the more space it can take up, spreading a little along the gradient. Once the proteins are focused to their isoelectric point, the voltage can be turned off and the fractionated proteins quickly harvested before they diffuse away from their isoelectric point, the pH at which the proteins are neutral. In this example, the sample has been fractionated into chambers of separate pH ranges, such as this pH 5.4 to 6.2 in this example. You can clearly see in this example that an increased number of proteins can be visualized after fractionating by pH. The entire unfractionated sample can be seen in this 2D gels, where the proteins have been separated by charge in the left to right direction, and then by size in the up and down direction. If we separate by charge first, and then take the liquid from this chamber, which contains proteins whose charge is neutral between pH 3 and 5, and we separate them on a 2D gel of the same size but a reduced pH range, we can see that this group of proteins with about 6 spots expands into this group of proteins of about 30 spots. A similar increase in proteome coverage happens with other pH fractions, greatly increasing the information that is obtained and the number of spots. Fractionation methods are often combined into a sequence to reduce complexity, and the order of these methods depends on the individual researcher and what else is present in the sample, such as SDS, which prevents charge-based fractionations until it is removed. One important point to make is that fractionation can be performed on either proteins or on peptides made from those proteins. Peptides are generated by enzymatic digestion of the proteins, and because they are smaller, they are more likely to be soluble, which is especially important if the researcher prefers to perform their fractionation and analysis by chromatography, as in this example, rather than by electrophoresis. While it is well documented in the literature that analysing the proteome through its peptides rather than its proteins can increase overall proteome coverage, in other words, it makes the list of proteins identified longer, it often comes at the expense of lower individual protein sequence coverage, where the presence of a protein is inferred by a small number of peptides being detected. This topic deserves an in-depth analysis all of its own, and we won't do it right now. The point that is trying to be made is that proteomes are really complex. A proteome can contain between 1,000 and 25,000 different genes encoding for different proteins, and these can be produced at as little as 1 molecule per cell for one protein and at millions of molecules per cell for a different protein. In addition, different molecules of the same protein can be modified in different ways, and these are not predicted by the gene sequence. Analysis of the proteome can be undertaken using different methods and technologies that give similar but overlapping answers, but with distinct information that is not provided by other techniques. The biggest challenge when analysing a proteome is an experimental design and thinking about what methods and technologies should be used to answer the biological question that is being asked.